Good morning, everyone. Good morning again. Yeah, hi, I'm Dennis Sikowski. I'm the Science Director here at the Hudson River Foundation, and welcome you all again to our first Tuesday seminar series. And for those, we have about 20 people here in the room today, and we have a nice audience that's on the webinar. I think there's about 50 or 60 of you that are viewing this on your computer terminals. We just want to go over a few ground rules, particularly for those people that are on the webinar, that hopefully you can see us and you can hear us. We can't hear you, but at the end of the talk, we want to welcome all of you to ask questions, and you can write the questions on your screen, and we'll be happy to read them off, and Dr. Lohmann will address them. Melissa from the foundation is standing by, so if any of you online have any issues, you can send her a note if you haven't already, and we'll try and get it fixed up. Okay, so today we're very pleased to have Dr. Rainer Lohmann from the University of Rhode Island to chat with us today about some work that he's been doing in Passaic River. There's a lot of interest, obviously, in the Passaic River. It's near and dear to our hearts. It's been near and dear to a lot of people over the years for a variety of reasons, used as a recreational resource and sort of a dumping ground for a variety of things. We're sort of pleased that there are some independent scientists that are out that have an interest in persistent organic contaminants. And we've been fortunate here at the Foundation now on, I think, 2 occasions to fund some of the work that Dr. Lohmann is doing. He has an interest in fate and transport of organic contaminants worldwide. He's developed some very, very interesting sampling techniques to more readily quantify a variety of persistent contaminants. Currently, he's been looking at not only the movement, transport, I guess, and the relationships of dioxins and other contaminants between air, water, and sediment, but also looking at the transfer of these things into some of the critters that live in the Passaic River. So I know I'm very interested to hear about the results of some of the work that's gone on over the last couple of years. So without any further ado, Dr. Lohmann.
Thank you very much. Welcome for those that are here and those that are on the web. So yeah, I was funded by the Foundation, I think 3 years ago now, to look at different organic pollutants in the Passaic River and the food chain. And it was a collaboration of 3 academics. There was Mike Weinstein and Kirk Barrett, who at the time were at Montclair State. For some reason, that is not a place we want to stay. So one is now at New Jersey Institute of Technology. The other moved to Manhattan College. And then at URI was my postdoc, Mohammed Carey, who did most of the work, and you'll see it's a lot of work. And Dave Edelman, who's my lab and field guru, who helped us with all the technical sides of deployments. And that's where we ended up working along the Passaic River. I understand that most of you are just interested in the dioxins, which is somewhere here. No, there. Sorry, no, got it almighty there. There we have them. But we look at a range of compounds that includes the old pesticides, new pesticides, PCBs, pHs, dioxins, and some other stuff in general. All right, so what I wanted to cover this morning was first look at contaminants in sediment biota, so very classical sampling of sediments at the surface, catching fish, and then analyzing them and looking for pHs, flame retardant, PBDEs, and dioxins and furans. And the second part is where we're using our different sampling approaches, so passive samples to look at what is truly dissolved in the water, what is in the gas phase in the air, and that helps us to establish air-water exchange along the river. And then the third part was to look at different necton, I guess they're called, or biota, that were caught, hosted by Mike Weinstein along the Passaic, and to see what is present in the different trophic levels and what does it tell us about the pollutants and their mobility. And it was going to be a really relaxed academic style talk, you do some science in a polluted place and there's no consequence to any of this. And then, 2 weeks ago, so EPA decided to unfold this plan to remove basically all sediments that ever existed in this river. So, I guess I'll have to say something about it. Luckily, I don't work for EPA. I don't have the legislative hoops to go through, so it's just an academic perspective. Keep that in mind. So what they're going to do is remove what is going to be one of the biggest cleanups in history, around 4 million cubic yards of contaminant sediments from the lower 8 miles. And let's see. So this is where we collected sediment, and this is the area to be dredged by EPA. So we have a pretty good overlap, because we had a high coverage of the lower 8 miles, of course. We had a little in Newark Bay, and we went a little upstream, and then we had one sample that was above Dundee Dam. That was supposed to be our reference site, because at least we assumed any dioxins couldn't move up the dam. So that's going to be our cleaner reference site, and we'll see how well that worked. All right, let's look at the data. So this is concentration of polycyclic aromatic hydrocarbons in sediments. So surface sediments, grab samples, microgram per gram, so pretty high concentrations. You see on the right Newark Bay. Then you see the arrow here is the lower 8 miles. They're going to be dredged if this plan actually goes ahead in the next decade or 2 or 3. And then I believe at mile 11 they already did some extra dredging, so that's the asterisk here. And then the highest point here is above the dam, so that would be our reference site. And yes, you can see our work would also suggest, yes, lower 8 miles are pretty bad. So no big disagreement. Maybe the mouth of Newark Bay is looking a little dirty. If we look for flame retardants, the picture doesn't change that much. Everything that changes is the scale here. We're looking at nanograms per gram, so we're 1,000-fold less. But nonetheless, you see them a little up the dam, fairly low at the higher part. It's certainly elevated through the lower 8 miles, and actually elevated throughout Newark Bay. So a little different story, maybe slightly different pathways. I'm not sure I know where they're coming from, but certainly they're very high in the same region where some of the other contaminants of concern are present. And so I was told you're all interested in dioxins. So dioxins, again, we have, so on the top we have dioxins, we have furans. Below, you can see, I guess you're interested in 2378 TCDD is striped. You can just about see it down here, little bars. And the most toxic furan, the 12378, is in this, whatever that is, turquoise. Again, obviously you see it highest around the Superfund side, the actual one, very high along these 8 miles, very low above, and certainly elevated, at least in the entrance to Newark Bay, and pretty much the same for the dioxins. So, yes, if I had to dig out somewhere, I guess 8 miles seemed a good bet. Just in case, because sometimes there was discussion where these are really from, this is very simple. It's a linear regression. It goes from the Superfund side up to the dam, and yes, we get a very easy fit, suggesting yes, it's really coming from that side and it's been transported out over through movement of the water. And of course, this is surface sediment, so concentration we find here are much, much lower than what you find if you go to depth. If you think a linear regression isn't a good fit, we can also do it in a more polynomial fit. It doesn't make a difference. So again, we have the Superfund site here, Leicester Avenue, and then we see the decrease to 17, 18 miles and into Newark Bay, and it works again for both total dioxins and TCDD. So I assume at this stage there's no more discussion where these are from, but certainly all the results point, yes, it was Leicester Avenue, and it's just been moving away from that site. We look at the toxicity which make the Superfund site so special. It's almost exclusively 2378 TCDD at 100, 200 picograms per TQ per kilogram per gram dry weight, much higher than the furans, and really 2378 covers almost all of it. So a very unusual signature, nothing you would find in any other places where you have emissions from incinerators or any other of those processes. So a very unusual sign, again, TQ-wise. There's a high concentration below 8 miles, very little above. and notably at the entrance to Newark Bay. So and then we also matched that with sampling of biota, and that is Mike Weinstein, who's now at the New Jersey Institute of Technology. So he did a combination of all kinds of different trawlings and nettings and maybe probably spear fishing. It was above Dundee Dam, was our reference site, in the oligo-mesohaline zone, which is the zone where there's very little salt, so the upper part of the river, and the mesopolyhaline, which is the most saltier lower part, close to Newark Bay. And totally got around 11 different fish species and blue crab, and we pooled them, so we ended up with 23 different samples. Because I'm a chemist, I don't know any about this stuff, so I thought I'd put a field guide in here who sent us that to explain what he found. I guess most important, there's the striped bass, which some people like to eat. So this would be the top of the food chain, we would think. We have things that just eat. On the benthic side, mummy chalk that eats omnivore, meaning it eats either fish or grass, basically. There's a carnivore, so it only eats fish. There's an herbivore, so it would just eat grasses or algae. And there are some others. So those were most of the critters that he found. And the blue crab. So they're a little slower, right? So that's kind of the most symptomatic species we got. So to just orient you here, this is a summary of all the results. So here the first 5 bars are above dam, so that's the reference site. And the next are the upper Passaic River, and then that's the lower Passaic River. Maybe the easy part is, so the middle are PCBs, and the lower are dioxins, and you see the results are pretty much the same. You've got really high values for the blue crab, and I'll get back to that later, but blue crab is on the top of the food chain, but it sits right on the sediments and eats a lot of that stuff, so I think this is really an indication of its diet, as in its location in diet, rather than that it's the striped bass, which is somewhere else here. Saxonitis is here, so that top of the food chain is high, but it's not as high. And we have basically same concentration throughout most of the Passaic River, and then just lower above dam. pH is a little spottier. pH results were actually interesting. We'll get back to that in a minute. But so there's the food chain results, and you would expect. I mean, these fish, they're not as stationary, so they will move around. So you would expect that the signal wouldn't change that much throughout the river. The toxicity is actually interesting because it's still 2378 TCD that drives all the dioxin toxicity, so that's everything in blue. In yellow is toxicity by PCB 126. Very different uptake pathway. 2378 TCD is probably exclusively through sediments or the food chain. 126 is actually found in some of those species that feed on algae, so the lower trophic levels animals. The blue crab is here, and that's all sediment dominated. And most of these animals are dominated by the sediment food chain, but some reflect more the water column, and they're actually 126 is driving their toxicity. So it's a two-pronged approach for dioxin-like toxicity in the river. And that might be a little surprising. It's just 126 is present in that much higher concentration that it can outweigh or at least match the 2378. We look at the flame retardants. The reference site isn't as reference anymore, and PDEs are so widely used across homes and industry that it's probably not a big surprise. So the reference site isn't as cleaner anymore. The upper river is a little higher in concentrations, but that's partially because we have the crab here again and the lower river end. You see similar spread of results, but again, the Passaic River is on average certainly higher contaminated than other places. So this is trying to summarize all of that in a table. Hopefully it doesn't have too many entries. So what we have here is concentration of pHs, dioxins, the TQ, and PDEs. And this is above dam, the mile 8 through 18, mile 11 that was dredged separately, miles 0 through 8 that are going to be dredged, and then Newark Bay. So those two are basically targeted EPA, and for a good reason. The only thing I would say is if you look at the numbers, in terms of TQ, at least the entrance to Newark Bay doesn't look very different. Now you can argue if an industrial harbor should have the same quality and sediments as a river. I'm not going to get into that, I just point that out. You don't have to hold a harbor as clean as you might want to have a recreational river. The upper 8 miles, or about mile 8, they're certainly cleaner. So, as I said, I'm just a scientist. I'm not involved in all the legal stuff, but the actions that are proposed make a lot of sense. I'd probably go to Newark Bay a little bit and scoop out some extra dirt. I mean, if you collect 4 trillion, if you collect 4 million cubic yards, if you do 4 or 5, I mean, it doesn't make a difference, right? And I don't pay the bill. But so that's just from our work, and I think we basically see the same in the biota. The only thing I would add is, previously we worked in Newark Bay, and we had trouble finding biota. So it was very difficult. There wasn't that much living there we could scoop up. The Passaic River is a little easier. All right, so that was basically the classical thing where you scoop up things or catch something, grind it, extract it, and analyze it. In the second part, we used a slightly different approach to look at what is dissolved, and we wanted to know what is dissolved in the water because that drives bioaccumulation only. That's how things get mobile, how they're mobile and getting into compounds, so getting into biota or getting into the air. So what we used are these plastic sheets that she holds here. So it's a strip of polyethylene. It is an industrial material we buy at Home Depot. When we put it in the water, or in the pore water and the gas in the air, compounds move in for diffusion. And that gives us one big advantage. We don't have to worry anymore about colloids, or what is bound to particles. We only measure what is truly dissolved. And that makes it a lot easier if you want to look at bioaccumulation or air-water exchange. The flip side of that, we have to know how this sampler works. We have to know something about the partitioning into this sampler, the K value, and how this works at temperature, salinity, and how long it takes to equilibrate. And once it is equilibrated, we know what's in the passive summary, we know the partitioning constant, then we can divide the two and we get the dissolved concentration. And in a few slides I'll show you some of those concentrations. Maybe some background why these were developed. And I think the first and obvious point was that we know now that sediment concentration, which I just showed you, is not a good proxy for bioaccumulation. There is no real link between the total concentration. Something's falling apart here.
Yeah, I'll put that together for you. Keep talking.
I guess by the end of my talk, this whole building will collapse. All right, so total sediment doesn't quite work. Equilibrium partitioning, which was used to convert sediment concentration to bioaccumulation, also doesn't quite work, at least not if you just look at organic carbon. And so what was finally developed over the last, 10, 15 years were passive samplers, which give you a direct measure of pore water, and that indeed then drives bioaccumulation. And there's this truly dissolved or pore water that can also use to assess the transfer into the food chain. So the earliest design, and some of you probably have used this, is this SPMD. That was a geochemist view of a fish. It's a membrane that has lipids inside, and that's how we would think of as a fish. That was what Jim Huckins and USGS developed. And because it has these lipids inside the plastic tubing, it almost works as an infinite sink gas, and it never equilibrates. It could out be there for a year, and it would probably still take up contaminants. Now, these were patented, and they made it a little expensive. Thank you. So, over time, people have moved away from these complex, very simple samplers that have just a single phase, and we use the polyethylene, which was what the LDPE was, what the SPMD was made of. The Dutch use silicone, others use polyoxymethylene, others use spiny fibers. I mean, they're all different approaches, but they all give the same result. They give you a truly dissolved concentration. In our case, we stick to the polyethylene because we work with them enough that we know how to use them in the gas phase and for the dissolved phase. And that's what we're going to show us. One last word. The other reason we'd like to use them is if we have to do active sampling, depending on where we go, we might have to collect tens or hundreds of liters to overcome detection limits, and that's logistically very difficult. Over time, electricity pumps, it's very involved with polyethylene. We put out a few grams and we get the same result. end up using a lot less solvent, and maybe what's most important, we often can actually skip the cleanup steps, so we have a much higher turnover in the lab, which also means we can produce more, and so my students or postdocs end up processing a lot more samples for the same price. Just a little background on how we actually do this. So we buy them and we cut them into the size we need, around 2 grams, probably a sheet of that size. We clean them a couple of times. We add performance reference compounds, and then we're ready to deploy them. So the performance reference compounds we add, so this is just an example. I have 3 deuterated pHs shown here. We know their concentration at the beginning, then over time they will leach out and then eventually reach 0 concentration in the sampler. And at the same time, our compounds of interest will start accumulating in. And at any given time when we take them back, knowing the loss, we can extrapolate to how far our compounds of interest are from equilibrium. We can correct that way, because not all compounds will equilibrate within a 1 or 2 month window. So the 2378 might be 60% equilibrated, OCDD only 15%, so we have to make up for the loss. All right, so these were our assembly locations. We had 6 sites where polyethylenes were deployed, and here also where the biota were sampled. Oh yeah, and this is a slideshow of the beautiful part of the Passaic River. So there are 3, these are the sites of deployments. There are 3 rowing clubs or boat clubs, and then there is a county jail parking lot, I think that's that one. There is a restaurant, backyard at least, and I think there's a major bridge. And these were all, you can just about see these stainless steel bowls, so that's where the atmospheric sampler was taken, and then somewhere in the adjacent river bank was the water sampler deployed. So those were the sites that Kurt Barrett found for us and operated year-round. So I'll give you an idea what the results look like. Looking here at nanograms per liter, 100, 200 for the pHs. So this is the reference site above dam, and then we go from mile 12, mile 9, 6, 4, and then in Newark Bay. And so you see our reference site isn't ideal in this particular case because it's actually much higher than most of the river. Newark Bay again is pretty high. Now pHs come from all kinds of processes, including direct spills of oils, traffic exhausts, you name it. So you would expect an industrial harbour to probably have high concentrations. It might be not a big surprise. What I'm trying to show you is just a very simple comparison of the trends that we see in the water, as in here, to what we see in the sediments, and that was that profile. So I think the only thing I would say is that there's no concurrent trend which might suggest that sediments are not the sole reason for the pHs that are dissolved. Of course, there could be stuff coming in from air, and there could, of course, be all kinds of other sources that be that discharge from wastewater treatment plants, stormwater overflows, industrial activity. So there are all kinds of other possibilities that might explain this particular profile, which certainly does not suggest that the Passaic is causing contamination of the Newark Bay. But I guess for pHs, you wouldn't expect that either. This is a little busy, so let me just explain. At every site, we basically have an annual profile. Every 2 months, one sample. So it goes from November to February, April, June, July, and then back down to November. And in the black, the squares, you have the temperature, and then you have concentration of the pHs. And as a very simple first approximation, if sediments are a main cause of concentration, would expect that concentration will spike in the summer when temperature is up and biological activity is up. Then you have bioturbation, and that will normally release compounds from the contaminated sediments. And the only thing that seems for certain is that a lot of places there's either no correlation or anti-correlation, making me think that sediments are not the sole cause in this particular river for what is dissolved. So some of the dissolved compounds are from somewhere else. At least that would be my guess at this stage. If we look on the gas phase, so in the atmosphere, profile is again slightly tilted towards Newark Bay, again in view of all the activities. That's probably not a big surprise. Knowing now the gas phase and the water, we can, for the gas phase with the same annual cycle, and here you can see slightly better correlation with temperature. but it's not very strong, but it's certainly present. We'll see that better later on for dioxins or flame retardants. All these compounds volatilize from contaminated soils or water bodies, so they always are high in the summer and low in the winter. And that's certainly seen to some degree for pHs, suggesting there's some degree of volatilization going on. All right, having now gas phase and water, we can look at air-water exchange. And we looked at a range of pHs. So the left bar are naphthalene and small alkylated pHs, so those would be from petroleum basically and combustion processes. And there it goes either way. Everything going up is volatilization, everything going down is deposition, and we see it going either way. And fairly high fluxes, looking at 10,000 nanograms per meter square per day. On the right-hand side are more of the classical pHs, so fluorantine, pyrene, and for those, by and large, they're all volatilizing. So, however they get into the river is much higher in concentration than the overlying atmosphere, so the river will volatilize those out on a regular basis. We're looking at around 1,000 to a few thousand nanograms per meter square per day. So fairly significant fluxes, of course, on a small volume or surface area basis. This was my 4.2, so close to superfilm. This is Newark Bay, and you see basically the same trend. Most of the light pHs, we get fluxes either way, tendency to volatilization, and for most of the higher pHs, just in volatilization. So the irony is that, of course, we're close to Newark International, which presumably will affect air quality a little bit, but it seems that Newark Bay has sufficient pHs that they will volatilize out even in Newark Bay. All right, so on the dioxin side, again, what you see is total dissolved concentrations. This is our reference height in mile 18, and then going down river. Concentration gets well highest around mile 5 to 6, and then decrease very gently. So fairly well mixed river. And again, the first very simple idea was to look at how does the sediment profile look, and to the best of my knowledge, they kind of match each other. So it goes from a lower towards a higher and back down. I should point out that there's a little mismatch between compounds we find in sediments. A lot of the higher chlorinated ones, the compounds we find dissolved, are mostly lower chlorinated ones. So you have to keep that in mind when you do the comparison. But at least it doesn't disagree with the notion that there is influence of the sediments. If we look at the profiles over time, again, this goes from winter 2011 to the end of 2012. As temperature goes up, concentrations go up and then go back down. And basically this kind of works at most sites, which is in line with a situation where during the summer things get warmer, the mud gets stirred up, and you have release. I should note though, so let's say the small green here is 2,7 or 2,8 dioxin. That is not a toxic one. That's dominating. That is probably a byproduct from triclosan. The antibacterial, another major player here is, again, a not toxic dioxin. The 2,3,7A is in this dark red, and it shows up here, Maya 4.2, so close to where the Lister Avenue Superfund site was. So yes, you see it in the water column that is released, especially when it's warmer. And of course, that'll all change once EPA gets its will and dredges everything away. If you look at the gas phase, as you would expect in the atmosphere, it's fairly homogeneous. Highest concentration above Newark Bay. I think we saw that in previous work too, and that's mostly because of this triclosan derivative. So it's not linked to the toxicity of the Passaic River. If we look at two of the seasonal cycles in the atmosphere, we see a fairly strong gradient where it's lower when it's colder, goes up when it's warmer, and back down. And we see, so this is mile 18. There's no 2,3,7A TCDD we can find because it's so far away, mile 4.2. See about this is here in this dark, whatever it is, block. We see a little bit of it, but not very much. So, because there's so much 2,3,7A TCDD in the sediments and the water column, we see a little bit getting into the atmosphere, but it's a small amount. And if we do then the air-water exchange, it's not a big surprise. Obviously, almost all compounds are volatilizing. This is mile 4.2, and here is the 237A TCDD signal, so yes, a little bit gets out. And hardly any of the other compounds, because they are so hydrophobic, they bind to sediments, to particles, you can't get them into the atmosphere. You get them into the water column a little bit, you get them in animals, but they don't really volatilize out easily. And most of these are obviously the small chlorinated compounds that EPA wouldn't care about. But we're an academic lab, so we care. Lastly is the flame retardants. Again, the dissolved concentrations, a little higher, the reference height, and then going up, fairly steady. Right. It's funny, this graph has been giving me trouble for days now. This would have been the sediment profile. It kind of goes like that. I could draw it on the screen, but I might not make friends that way. All right, you've seen the sediment profile before. It's similar in shape. If we look at the concentrations that are dissolved over time, so with temperature again, it's pretty much the same cycle, maybe best seen mile 5.5. Very nice correlation with temperature, similar, I'd say, 4.2. Again, it's in line with the situation where they are remobilized from the sediments over time, over season. In the gas phase, we now see a slightly different trend. So PBDEs are correlated where people live, with industries, wastewater treatment plants, which often correlate with people again. So we now see our reference site, which might actually have more people living there, has highest concentration, then it kind of goes down as you go towards New York Bay. The gas phase profile is classical, really low when it's cold, gets really high and goes back down. I think Ron Heitz deduced somewhere here, July 17th is PBDE day, because that's when everywhere concentration is highest in the US. And I think we're close to that same numbers. And in terms of dominating here, will be really low brominated, PBD2, A15, they drive most of the gas phase concentrations. The more industrial, here you get some higher brominated ones. And if you do the air-water exchange, either at mile 4 or at New York Bay, very strong volatilization. These are not very volatile compounds, so they come through the water and then get into the atmosphere. And we see very significant fluxes of few thousands picograms per meter square per day. So, does the sediment drive water concentrations? For pHs, I'm not so sure, because there was no obvious trend in sediment concentration and what is dissolved. And of course, I started by saying that sediment concentration doesn't equal accumulation or bioaccumulation, so that doesn't mean that there is none, but it's not as obvious. Certainly bioturbation doesn't seem like a strong source. There are probably other ways in which they get into the river, but we still have mostly water to air transfer. For the flame retardants and the dioxins, we see very similar trends as in where the sediment is high, water concentration tend to be higher. We have a temporal trend where concentration of the water are highest during the summer, which would imply bioturbation of contaminated sediments. So sediments are an obvious source, and certainly we see across all of that river that the water is more contaminated than the atmosphere, so we have volatilization throughout. And for the gas phase, we see very strong temporal trends, which is very typical. That's a pretty good sign for us. And Passaic River is certainly one, but not the dominant source for the atmosphere, because it's a small river after all. So any other sources would probably outweigh it for concentration of the gas phase of almost all of these compounds. The only exception might be the 237 TCDD, for which we don't have many sources left anymore. And then a really contaminated river becomes an obvious point source. So I guess summary is, yes, from what I can see, the sediment is probably driving a lot of the water concentrations. Okay, and then the last part was to look actually what happens in the food chain. So this is where Mike Weinstein became very essential, because I'm a chemist, I couldn't tell one from the other. So there are a few concepts that we explored here. And the first one is a very simple bioaccumulation. That's the increase the concentration of the lipids divided by what is in the water. And that just tells you how much more this compound accumulates relative to being dissolved. But that's just the first step. The second then is, can we find evidence that compounds magnify? So that means they accumulate beyond equilibrium partitions. So they increase much higher than they should be based on thermodynamics. And if they do this really strongly, they biomagnify. If they do it poorly, they can actually biodilute, as when they broke it down within the organism. And one way to address this is a fairly recent approach to look at the trophic magnification factor, the TMF, and that employs the slope of a line of lipid concentrations versus trophic level. And I'll show you some of those graphs and then it all makes sense. So the first thing was to establish where these things live. So we used stable carbon isotopes versus stable nitrogen isotopes. Nitrogen is good for identifying trophic levels. Those are kind of indicated here through the bars. So at the very bottom of the food chain we have primary production, so phytoplankton. So that will be trophic level 1. And we have zooplankton grazing on them, that will be trophic level 2, and then small fish, big fish, and then polar bear or human on top. That's kind of how this works. So the two species that are of interest here is the crabs are somewhere here, so they're not really high up in the food chain. And way on top is the striped bass. And any of you who's fishing, that you would know all of this. And then all the other animals are somewhere in between. But we can position them through the use of these isotopes. And then we decided the trophic level as a very simple approximation. We used fundulus as the base, 3 per mil increase for nitrogen 15. That's how we came up with the food web, or Mike Weinstein. So we had the minnows, mummy chops, killifish, shads. White perch is already above them, and the striped bass is the top predator in this particular river. So anything that bio-accumulates very strongly should be highest in the striped bass. And if they're the same in all of them, then there's no biomagnification, there's no strong bioaccumulation either. Alright, so these are pHs. Concentration is the bioaccumulation factor, so how much they accumulate. It's any from 10 to the 3 to 10 to the 6. But there's absolutely no correlation with liquid water. So a bit of an odd result, and we'll get back to that in a second. pHs are known to be metabolized in higher trophic levels. So we didn't actually expect to find them I didn't expect to find them in most animals because I thought they would be broken down. I think because concentrations are so high, the animals didn't really have a choice, and their metabolism couldn't keep up with keeping them out completely. For PCBs, this is more the results you would expect. They are not broken down quickly or easily by most high trophic level animals, so you see an increase with increasing hydrophobicity, so you go from 10 to the 4 enrichment in the lipids to 10 to the 9 or so, so very strong accumulation, and it's roughly a linear relationship as you go up in hydrophobicity. For the flame retardants, we see the same. There's an outlier here, PBDE 99. I'm not sure if there's an artifact from our lab. Again, a little bit surprising to me because we know that animals can debrominate these. So in the liver they can detoxify them. But again, it might just be a question of kinetics, that uptake is faster than elimination. And of course, the poster child of the Passaic River, wonderful dioxins. We see by and large increasing accumulation with hydrophobicity. There are a couple that are way below. These are all the lower chlorinated ones, which can be detoxified in the liver. So they don't accumulate as much, and you obviously see that already here. Right, and then we want to look at this trophic magnification. So this is the extent to which they increase in top predators beyond just simple equilibrium partitioning. And for the really small PCBs, the slope of this line is negative, they actually biodilute. So the animals can basically decrease the concentration through metabolism. But if we look at some of the higher chlorinated PCBs, these are of course the very stable ones, 2852 all the way to 180. We see for all of them that the lines are going up. So this is actually an increase. And we have trophic magnification factor from 2 to around 3.4. I think the maximum that has been observed is around 4. I don't think any compound has been seen that can exceed roughly 4-fold increase per trophic level. So this is strong evidence that they all biomagnify. Now I should point out there's 209 PCBs. Not all of those persist in animals or in humans. These ones have been chosen to represent PCBs because they are so persistent. So there's some inherent logic here, a little bias. We look for dioxins. Again, there are some that decrease with increasing trophic level, and these are all the lower ones, and that's why they're not on EPA's target list, because we know they don't accumulate that much. So there's a 2-chlorinated dioxin, dichlorinated, trichlorinated, and we see a decrease, so that there's dilution along the trophic food chain. But then if we look at 2378 PCDD, the 2-chlorinated furan, and the pentachlorinated furans, we see strong positive lines and trophic magnification of 2 to 4. So again, they reach maximum increase in the food chain. So at every trophic level, concentrations increase strongly. And that's exactly for this reason that they are banned by international treaties. They're on everybody's target list and on EPAs in the US and in all the other industrialized countries, because of these very properties. So in a way we confirm what we thought was going to happen. If we put them together, seem to be a kind of curved relationship. This is trophic magnification. It starts to show up at around log KW of 10 to the 6, then is high up to 10 to the 8, and then seems to drop again. And this was postulated before for bioaccumulation. It seems to be there's some steric effects. If you're much beyond this range, the molecule just becomes very bulky. It seems to be a problem of getting compounds in, so that might limit to some degree the transfer. If you're much lower, it seems to be the place where metabolism is fairly fast. So we even see hydrophobicity to some degree influencing how much they magnify. All right, so we may wrap this third part up. So pHs were surprisingly high across all trophic levels, but particularly in the lower ones then obviously. And we saw few of the PCBs and dioxins can be metabolized, so we see biodilution. But we see strong biomagnification for most PCBs and dioxins, furans that we worry about. So if I go back to what I wanted to cover, first we're to look at all the sediments and the biota, look at what is present, and look at what the dissolved compounds in the air and the water, air-water exchange, and look at the food chain. So maybe rephrase that. I guess if I was in charge, I guess I would dredge the entrance to Newark Bay, just because. And from what I can see, there's obviously good reason for dredging because the sediments drive dissolved concentrations, particularly for the dioxins and the PBDEs. And I think we have the same results for PCBs, but we were running out of time to get everything together. pHs were high in the lower trophic levels, and the dioxin, the PCB and PBDEs are higher in the higher trophic levels, which is exactly what you would expect for those persistent compounds. And yes, the one thing I didn't discuss, but we excluded the blue crabs from all these food chain studies because the concentrations are highest everywhere, but it's not because of their food chain position. I showed you in the big plot there was somewhere in the middle, but it's because they just eat the sediment all the time. And that's a really dumb thing to do in the Passaic River. So they have really the highest concentration you can get. All right, with that, I guess I'll thank Dennis Foundation for funding me, you for coming or listening, and if you have questions, let me know.
Questions?
What would you expect to find in herons and gulls? Fish are very, herons will eat blue crabs and all sorts of things off the pond.
Yes. No, you're right. They often show up in the food chain studies. They show up higher trophic level and often at very high concentrations. just because they're so mobile that it gets very difficult to assess where they eat all the time. So if there was really heron that would just be eating the Passaic River, it would go up even higher. But because they often fly around, it gets very difficult to, it might be in the Passaic, it might go somewhere else, it might go to the clean New York harbor, it might go somewhere else. So it gets tricky. It might fall off because of that reason. The other problem is that you go from animals that are cold-blooded to a warm-blooded animal, and that makes the next step more difficult to assess. But normally their concentrations are way higher. So, yes.
I might suggest that the striped bass, also migratory and not necessarily the best higher trophic species. They'll go into the packing sector of this for a few months and then go out to the open ocean. And they don't originate in that system either. They just come in from Hudson.
Yeah, no, it's a good point, and I didn't go into the detail. We realized the same problem. We had 3 size classes of striped bass, and the smallest one were much in line with the food chain model that would make sense. The 2 higher ones you could see from the isotopes that they had eaten a lot of marine stuff. So then the contaminants, basically the contaminants that they might get from the Passaic or Hudson get diluted as they move off. Sure, and we caught them back. So yes, they're not ideal. I guess that's why there aren't a few studies in river systems or estuarine systems, because of that dynamic. So yes, in this particular case, we included all 3 striped bass, but yes, it's not a perfect target. Fully agreed.
Could you clarify, you said the blue crab concentrations were the highest, but they're low on the trophic web. Are you saying that they're not important for prey? Because of the biomagnification that you
And if you could try and repeat the question, Reiner.
I'll try. The question was about the blue crab. So the reason we excluded it from the food chain, from the trophic magnification factor, is just because the reason the concentrations are high in the blue crab has little to do with their trophic position.
But why are the concentrations lower?
Because blue crabs have a direct intake from sediments, I think, that the other animals don't have. So their contamination is due to the way or where they live rather than what they eat. Well, I mean, they eat. It's not just the other fish eats specific things. That might be either a small fish, it might be an algae, or something like that, and that determines the trophic level and their contamination levels. The blue crab also, I think, takes in direct sediment, and that makes it much more contaminated than anything else. So that's why we took it out of that particular food chain model.
What was the size of the blue crab?
Ooh.
I looked at it.
I have to pass, because they were collected for us. I assume they were small ones.
Question of the sites you guys had to investigate. How far down in the sediment at the sites did you go?
For the sediment sampling?
Yeah, the sediment sampling.
So these were surface grabs, top 5 centimeters.
Oh, just
So yeah, the very recent stuff.
So we don't know. Has anybody ever gone further down, like particularly at 2 sites? The incinerator in Newark, and I was looking for wastewater treatment, but I don't know the sites in Passaic where wastewater treatment comes out. Because most of the Newark treatment comes out into the Hudson. The outfall to the Hudson. What the Passaic Valley Commissioners decide. So I'm wondering about the incinerator in Newark.
So, and this study, so the question was if we went to deeper depth.
You know, some relationship
Sure.
between the discoveries and the incinerator.
So in this study, we didn't go to deeper depth. In a previous study, we did, and you find that, well, we went around the Superfund side to deeper depth, and concentration were a lot higher.
So, and also the fact about the industrial area there in Ironbound, which is part of the Superfund site, would that fit into the pattern that you guys have discovered along the river?
I mean, even my postdoc looked through the profiles, and you can distinguish different source categories that would deliver dioxin to that river, of which incineration and other reasons.
Yeah.
And depending on what you look for, you get very different answers. But what is clear is that the toxicity is driven by that Superfund site, the Lister Avenue. That drives toxicity across all of that river. Total dioxins is a different story. So you get incinerated, deliver a range of dioxins. Some of those you can find. So it's not the only reason for dioxins, but it's basically the sole reason for the toxicity in the river of dioxin lights.
Why don't we take one of the, there's two questions up there, Reiner. First one from Melissa. Maybe you just read them off and
where sediments sampled in cohesive areas over the full length of the river, where upstream sample is more sandy. All right, so here's the catch that we work as a team of investigators. So Kirk Barrett and my field guys got the sediments. We covered basically the full length of the river. I mean, we had, 12, 15 sites along. Where upstream sample is more sandy, I do not know. But certainly will, once we process data further, we'll normalize to organic carbon, in which case it doesn't really matter anymore. So the answer is, I don't fully know this technical question. I'm sorry. Melissa asked about how fish and crab were analyzed, whether whole body samples or only fillers, edible portions. I guess this is the problem when you rise up in the food chain in the academia. You get removed from the lab side. Now I've got to think about this a little bit. 95% certainty, I assume we used fillets. I'm not sure for the really small animals we got, because we had so few of them to get enough mass to analyze them. But as far as I'm fairly certain it was mostly fillets. Now haptopancreas for the crabs. Oh dear. I assume it's included. That's my guess at this stage. So yeah. We're always trying to figure out, are we doing this for assessment of human risks, which would be fillets, or do we do it for the food chain where the animals just eat everything from the other animal? My guess is, let me rephrase this. I assume we analyze mostly the entire fish, because we're interested in the food chain, so in which case, if fish eats the other fish completely, they don't remove anything. I think that was our reasoning for also in the crabs to include everything. I can get back with details once I consult with my lab gurus. Any other questions that I can answer?
I have two questions for Professor Lohmann regarding the PCBs, particularly PCB11. The first question is, what's the pollution resources? Any combined solar overflow or wastewater, and also discharge of wastewater influence to the river? The second one is the PCB mostly analyzing water sample or sediment sample.
All right, so the second question I can answer easily. We analyze PCBs in all the matrices, sediments, water, air, animals, but we don't have all the results yet. So that's why I only showed a few. First question, PCB11. It's supposed to be mostly from pigments or from paint. There's a very unusual source. We haven't looked at where exactly it's highest, so I can't give you an answer yet.
Is there like a wastewater treatment plant on the river and potentially to discharge ground sewage in the river? What's the limit or something like that?
I think the biggest wastewater treatment plants would, and we had this discussion before, would discharge into Newark Bay or into the kilns and then would be flushed in from the bay, but we haven't looked at our results in enough detail to see if that makes sense. Sorry.
I just had a couple more questions about the data. So the only non-convergent issue you have are the graph?
Yes.
Right?
Yes, we only have.
And did you do any stomach content analysis for the fish?
No.
No.
No, we relied entirely on the isotopes.
Okay. And then the last question is, were they grab samples of sediment?
Yes.
I have a question. Jim, could you put up the sampling map again? I just had a question when you're referring to Newark Bay, and was that one, or is there a couple pooled samples?
Let me see. It's a little further down. Where is it?
When you say...
Oh, no, sorry.
Yeah.
Here. Yeah, so there were 4 samples around Newark, the upper part of Newark Bay.
Okay.
And that is the one that was fairly high, but it's obviously it's the outfall of the Passaic, so you would expect, just because Newark Bay, the contaminants won't stop. So this is the one that was basically as high as anything up here.
And where does the Superfund dredging start? Where's the lower end? Who knows the answer to that?
Yeah, pretty much right there.
Right at that point?
The mouth.
Okay. Yeah, I don't think you showed variability within those 4 stations. I'm assuming the one near the river, near the Passaic, you saw higher concentrations, I think.
Yeah. So anything that I showed in that table there, the highest one is always that. So anything on the right-hand side, the maximum concentration is that side close to the mouth of the Passaic River, not a big surprise. And but in the dilution, so the dilution to the other side, and you see the range is pretty big and the drop-off is fairly strong. So there's a very localized problem and then it dissipates.
Do you have anything to say about what struck me a little bit was that you are seeing a transfer of 2378, even higher chlorinated dioxins coming out of the sediment into the water. Do you want to say anything about transport out of the river further south?
Do I want to?
Well, I know you don't want to, but could you?
I assume, based on the previous work that you funded us for, it looks like the biggest transport is through sediments, then colloids, and then probably what is dissolved, just because the affinity for solids is so big. So yes, as long as you have water coming out of that river, you'll have transport of those compounds, but mostly bound to something else. You have a question?
Did you or the field people catch any carp?
I don't think so. I think they tried. I don't think they got any, but that might have just been bad luck.
Any other questions? Anybody else, Jim, on the written? No more questions? So we're going to be here all day. No one online has some more questions? Another question?
Well, just is there a report on this that's out there?
I'm sorry.
Is there a report available yet?
Is there a report available yet?
That is publicly available? I'm not sure. I don't know about your posting policies.
Well, the last thing I think we have is a progress report that if you want to release, we can make that available. But I guess the other question is, we've had some requests for the presentation itself. We'd like to post them on our website if possible.
You put a disclaimer.
You can put whatever disclaimer on it you'd like.
Yeah, sure.
Okay, and maybe given that, what would probably maybe another answer? When do you expect to be writing up some of these things?
Or yesterday? Yeah, the next year. you know how things are. I mean, we hope to have something out next week, and then it'll be next month. And then the reviewers won't like it, so it'll take a little longer. But so yeah, hopefully that'll be coming out in the next few months to the next year. That's my guess. It's just we have a lot of data, so it takes a while to wrap our head around all the details. And certainly some of the questions and discussions help us realize what we should make sure we get right.
So one final question. I mean, what would you consider the biggest surprises that you discovered?
Personally, I was surprised that you can actually cut the river into two parts, and only one part is really bad, the other part is not quite as bad. So that's what EPA is trying to do now, and I thought by now the river would be completely, with the tides moving in so frequently, I thought the whole river would be lost cause, but it's not quite as bad.
Anybody else?
Where are they collected, your water samples collected from a discrete location in the water column, or are they?
Yeah, they're normally around they will be submerged all the time. Normally I'd say a meter deep, but in the Passaic that's not always easy to do.
But they're not near the sediments.
No. From what I remember, that river is not always very deep and strong, so they'll be always in the water. I mean, if there's resuspension, they would see the particles, but they only measure what is dissolved. So we have pore water concentrations about ready, and that would be much, much higher. So we didn't sample sediments, and we can see that difference very strongly.
I guess that's a wrap. Thank you very much. This is our last seminar for the spring season, and so we're putting together something for the fall. And if we've got some requests from folks to give presentations, if any of you have any other suggestions, possibly for speakers, please talk to me afterwards, or send me an email, or just call me. And hopefully in the very near future we'll be putting out another schedule, and we'll be posting this on our site with any suitable clarifications. And also